Thank you very much for the introduction. It's a pleasure to be here. It's been really 2 enjoyable days. And today I would like to take you through our journey in trying to understand the wave molecules, the simplest you can get if you don't get to the quantum world, which I will just mention in passing. But it's quite basic for biologists and philosophers. We will speak mainly about molecules. I will get into technology because I think that evolution is the best engineer, and we will get to how wonderful ingenuity was found with molecules. We'll discuss the role of molecular self-assembly in disease, and also its role, well, theoretical role, hypothetical role in the origin of life. I must thank Moshe Kol for the wonderful presentation, the first presentation in this meeting, providing Chemistry 101, Chemistry for Poets, which I try to teach my undergraduate students for about 1/4 of a semester, the chemistry of carbon, and they learned chemistry in high school, and still it's a challenge. But so, how do we get there? So we are interested in learning and being inspired from nature. And our approach, I think it's very nice to say it in this building. It's a reductionist, minimalistic, non-biased. It's a reductionist. We are trying to find the smallest recognition module in nature that allow sociology of molecules, of 2 molecules to interact one with another to form ordered structure. It's a minimalistic. We're trying to look for the smallest one. It's non-biased. It's not that we decide this molecule looks very nice, we will choose it. The other one is ugly, we want her. It's a non-biased approach, and we're looking for the smallest molecules in nature, and you know all about molecules. Moshe gave an excellent introduction that can form ordered assemblies. So the self-assembly, and I will speak about self-assembly, don't worry, it results in very interesting properties, mechanical properties, very rigid structures, optical, luminescent structures, semiconductive, we all know about a semiconductive industry, piezoelectric properties, all the properties that we know from physics, usually of non-biological or non-organic structures with organic building blocks. As I told you, our approach is non-biased, and it's very interesting, and I will connect it directly to things that you are familiar with, or some of them, how we get by our minimalistic, reductionist, non-biased approach to the same thing that evolution have reached. And here are a few vivid examples. One is the physical colors in fish, which is not real color, it's a physical color. These are photonic crystals. They allow only part of the light to be dispersed, and then you see the colors. If you put this fish in a blender, you won't get any color. It's just the arrangement of this structure. The ability of chameleon to change its color, and it's quite new understanding. So it's, again, photonic crystals of guanine, the molecules that we found to be the best self-assembling structures, reflectors in reptile eyes, nocturnal reptile eyes, and also all kinds of structures at the cellular level, carbon-based structures that can form this known as G4 quadruplexes involved with the structure of chromosomes. So what is molecular self-assembly? What is it all about? And so this drawing of molecules in Moshe's lecture, and we are interested in the way for sociology of molecules to interact with one another. So this is the chemical structure, but they could be presented in a schematic way in these colors. So this is the red, this is the blue, this is the green. How they interact with one another. Sociology of molecules form ordered assemblies by miracle interaction, non-covalent. She was mentioned it already. I want to get the meaning of non-covalent is that they're not changing the properties. They are staying as they are by all kinds of interactions that are weak by themselves, but the combination of them makes ordered assemblies. It could be just by piling up structure on top of the other. So the self-assembly is moving from this disordered structure into disordered assemblies. It could be between the interaction of opposite charges to form the structure. I think it's, even though it's quite understandable. And why is it so interesting? We, in technology, current technology, we mainly discuss top-down processes, in which we go to more and more smaller and smaller structures to make more dense processors, more ordered assemblies. The idea and the way that biology is making technology is by self-assembly, organization of the molecules to form ordered assemblies, and this is really our approach. And this is not science fiction; it's reality, because we can see such structures. So Moshe again said about mentioned the Nobel Prize of making all kinds of machine, but biology is making even more complicated machines. Anyone knows what it is? A part of Moshe Kol. It's the ribosome. The ribosome Nobel Prize Ada Yonat a few years ago. This is a very complex machine, and all its size, it's much smaller. I will show later a chip of Intel. So all its size is much smaller than the resolution of the best microprocessor that we have, and it makes very complicated activity. So this is a machine. It's a machine that is built by self-assembly. It's not miniaturization of anything, not going to smaller and smaller parts, but by self-assembly, more complicated than this process, you build a very complicated machine. And we know that by self-assembly we can get all kinds of structures with very notable properties, by a spider silk, and with a popular culture, Spider-Man is known for the silk is very strong yet light. And indeed we know that we can make this technology come through, because we have billions of nanomachines in any drop of water if you have in the level of bacteria, and making things like motor, like shafts, all the things that we know, but much, much smaller. This is a virus. It looks like you have a machine that is, the size of it is only few nanometers, few tenths of a billionth of a meter, but still it's a very elaborate functional machine. We can make all kinds of things. Just to give you the reference. So we speak about 22 nanometer processors and 14 nanometer processors, and things are very elaborate and took many years to get it. But in a very simple bacteriophage, it's a virus of a bacteria. You see all the size with all these fine details is 5 nanometers. So by and all is done by self-assembly. By self-assembly, we can get ordered structures with unique properties, and we believe that understanding the way that molecules interact with one another, sociology of molecules, why one like one of the other doesn't like, how to separate them. This is the future, and I will show you a few examples of what we do with this. One of it is related to some mishappening in biology with neurodegenerative disorders, which is known as amyloid diseases. It goes back to more than 100 years ago, and actually the first instance was in diabetes. American physician discovers strange structures in pancreas of diabetes patient. The other one is much more across the Atlantic, much more known through the name. The German physician find in a patient with dementia deposits in the brain. what it has to do with what we discussed about self-assembly. Few tens of years later, it was discovered that amyloids, as we know, first of all called amyloid, are starch-like. It was very primitive at that time. The beginning of the 20th century, the way that it was done is by staining with iodine, like you do with a potato in kindergarten. But science was evolved to know that it's made by protein, and it's made by protein, thousands of protein joined together in a very unwanted manner, but in a self-assembly process to form the structure. Just like you see here, you have molecules joining together to form ordered assemblies. And this is the brain of an Alzheimer's disease patient as compared to healthy one. You see all these abnormalities. And what is understood now that you see in the brain all this very thin structure, fibrillar structure, between 7 to 10 nanometers, 7 to 10 billionths of the meter. Just to give you a reference, it's about 5,000 times to 15,000 times thinner than human hair. Blonde is thinner than black, just for you. If you get something from this talk, so a blonde hair is about a third of black hair. But to get serious, this is nanometric structure forming by self-assembly, and it kills the cells around in Alzheimer's disease, the things that are related to memory, learning and memory. In diabetes areas in the pancreas that relates to the production of insulin. And you see very long disorders in which you see the formation of these structures. One of the things that is related, it is common to all these disorders that are related to old age. So this is some mishappening of nature, the self-assembly. Unlike, let's say, cancer, that unfortunately we see also with children and with young adults, we don't see the same thing with Alzheimer's disease or Parkinson's disease or type 2 diabetes. But it's some kind of mishappening of of nature, and I won't get you into into our chemistry, but to make a long story short, in our approach to search for the smallest structures, we are looking for these red chupchiks that allows the formation of ordered assemblies, and then we can manipulate it and prevent the formation of this hair-like structure. And actually, we are very much involved in the development of, sorry for the chemistry, but I think it could be understood also intuitively. We're looking for structure, if you think about it as a Lego building block that bind on the one end but do not allow further growth because of this, what we call steric hindrance, because of the structure that we see here. And the most advanced compound that we have is already tested in humans. So this was one thing, and we were looking for the smallest modules that allow the formation of these structures. Based on, and with CAT, we were the first in the world to show that the pentapeptide, a peptide that contains 5 amino acids, this is the essential building blocks of nature, can form all the structures. And in our approach to search for the smallest structure, we identified, even if you're not a chemist, I think that you can appreciate this thing, which is a lecture, but these are quite simple molecules. Probably the simplest molecule, the peptides, fragments of protein that can form ordered assemblies from these nanotubes. Nanotubes are very much in the central interest in nanotechnology, the technology that deals with structures at the scale of few billions of the meter. It was published in Science, quite a good journal for those of you who are familiar with scientific publication, but also in the New York Times, and I realized that more people read the New York Times rather than Science magazine. And
the letters are larger.
Yes, this is the only reason. And we made all kinds of structures based on this. Already 13 years ago, we were able to make 20 nanometer wires, which are like the state of the art 15 years later, 13 years later with structures, coaxial cable. I just want to show you that we can play around at the nanoscale. We can make tubes, we can make spheres, we can make all kinds of architectures that we know in the nanoscale. In the macro scale, we can make in the nanoscale, all different architectures. And what actually that was discovered, and don't worry about the details, I will explain everything. We discovered that if you go to the most essential, most fundamental modules, you can get to very interesting physics. We discovered that by self-assembly, again, putting this structure, one on top of the other, without changing the identity of the molecules, we can take something that looks like protein and make things that are as strong as steel. I will give you also a mechanical engineering 101. This is the gigapascal, the Young's modulus is how strong is a structure. So cytoskeleton, what gives the cell its physical rigidity, is about half a gigapascal. The most strong plastics, like PEEK, they are about 4 to 6 gigapascal. Bone and teeth, they are about 20 gigapascal. Titanium, used for prosthetics, are around 100 gigapascal. Steel is around 300 gigapascal. Diamond, the hardest material, is about 1,000 gigapascal, 1 terapascal. So we could get by self-assembly things that are as strong as steel. We got, and then it's still a mystery. We collaborate with our friends at the School of Chemistry and the Weizmann Institute of Science. And this is, since we went to such small building blocks, now people are trying to explore how to study using quantum chemistry, the formation of the structure. So this is, well, it's even, I think, beyond some of my knowledge, but just to try to convince you to get to very small building blocks, you can get to very interesting physical phenomena as well. We see luminescence, nonlinear optics. I know that it sounds like buzzwords, but just to, I hope that I will convince you that we could get to very fundamental physical phenomena when we get to the smallest building blocks. Piezoelectricity, the ability to produce electric current by inducing force on the structures. Peptide and semiconductors, this rich nature, also a reasonable scientific journal. And there are many applications that, really a quantum jump, many potential applications. And I don't have a lot of time, but I think that I just gave a colloquium in Queen Mary University of London last week, and somebody told me that I'm speaking like a salesperson. I'm trying to convince you that these structures could be very interesting. Actually, published Nature Physics recently, we showed the ability to store mechanical energy in those structures, similar and being able to release energy very quickly. I think that the most the best example, which is known for everybody, is like the Venus fly trap, which must release power very fast, which means the power density. But our structure are about 3 orders of magnitude faster, faster than most of the structures known to man. Other people are using it as crazy thing. I have this Google alert on the things that we do. So people are using it as thermometers to measure, due to the optical properties, quantum confinement. I won't get you with all the details, but just to see how bizarre things like measuring temperature at 13 kelvins So it's minus 260 degrees. So going very, very deep there. People are using stent. There are about 150 groups working on these structures. This just published last month. There was people working on using the energy, using the piezoelectric properties, applying force, and you can see but writing the names of the peptides that are here. And so this was for this structure, and all of it is natural and probably the most fundamental building blocks in nature. But we are always flirting between the natural and the artificial. And as I said, now many other groups, especially in China, India, United States, are working, and we cannot compete with this anymore because there's a lot of competition. We decided to check whether we can go for the non-natural, and then this is, you heard Udi Shapira yesterday about DNA and DNA computing. Problem with DNA, it's a very complex molecule. He didn't mention it very specifically, but there's some kind of a hybrid molecule, which is protein on the one hand and have the structure of DNA on the other. And again, in a reductionist, systematic, unbiased way, we discovered that only, for the biologists or chemists, only G-containing, guanine-containing structures can form ordered assemblies at the nanoscale, which take us, again, it's the same guanine that nature is using over and over again to make functional structure in nature. And when you get new, and this is the structure, when you get new structure, you get new physics. Again, just on the tip of the iceberg is the strange physics that we see here is the fact that these structures could be of different colors. They can be blue, they can be green, they can be orange, they can be yellow, they can be orange. It relates to the wavelength of the color of the structure in which we shine them, which is a very interesting phenomena also from the physics point of view. Lastly, and here we provide some contribution. So over the years, we move more and more from biology to technology. And we ask ourselves, what is the smallest structure in our reductionist, minimalistic, non-biased approach? What is the smallest structure that can form ordered assembly? And we ask ourselves whether a single amino acid is the basic building block that builds proteins, peptides, and then proteins, whether a single amino acid can form all the structure. The answer should have been no, because for many years I would say that dipeptides, the one that I showed you before that got with all this crazy application, is the smallest one because there's some resonance around the structure. It forms two-dimensional assemblies. Give you all the chemistry jargon, but sometimes it's good to do to make controls. So this control would have been no, but we realized that a single amino acid, in this case phenylalanine, can form ordered assemblies, which in a minute I will explain why is it important. We scratch our head. What could it be? Could it be just another phenomena that we see? Or could it be a situation in nature in which you see such high concentration of amino acid that this structure could be formed? And we offered the hypothesis. In this case, I think that now the hypothesis is already proven, but it might be that the self-assembly of the simplest building block probably we can find in this molecular self-assembly process makes the formation of this fibrillar assembly, this air-like structure that we see with degenerative diseases, also in inborn error of metabolism disorder. Don't worry. Phenylketonuria. Think that anyone has seen this structure. When you read, if you are like me, you read what is written on every food product that you eat.
You can be a lawyer.
It's
Or a chemist.
or a chemist. Oh, well. So in Diet Coke, it contains phenylalanine. Phenylketonuria contains phenylalanine. What is phenylketonuria? It's a disease. It's a metabolic disorder in which the individuals they lack one gene, one enzyme that cannot convert one type of amino acid, phenylalanine, to another type of amino acid, tyrosine. So if they don't eat a very strict diet, they cannot eat any protein of any type. They can eat only very yucky type of structure that is based on carbohydrates and fats, and 19 amino acids. So, just to, as I know it's a tribute to Carmel, this is the friendly.
This is a peptide, in essence, a short protein. Our bodies are full of them. Some are useful, some are just byproducts of longer proteins being broken down. But under certain circumstances, when there are too many of them in the same place, they can band together and form aggregates. They are called amyloid formations. Those aggregates can be toxic to cells and are implicated in Alzheimer's disease, as well as in many other disorders. The conventional wisdom always was that only peptides built with at least a dozen or so amino acids, the building blocks of proteins and peptides, can form those organized aggregates. But scientists in Professor Ehud Gazit's lab have shown that amyloid formations can be created with very short peptides, and even with single amino acids. The aggregates the amino acids create look and act just like peptide aggregations, and are also toxic to cells. Could that be significant? High concentration of amino acids, like those that were found to create aggregates, is the cause of many metabolic disorders. For example, phenylketonuria, PKU, which affects 1 in 15,000 people. The amino acid phenylalanine concentrates in the blood and brain of the patients. There is no treatment. The only way to avoid symptoms, which can be severe, is to stick to a very restrictive diet. Since we now know amino acids can form amyloid formations, we can look at PKU and disorders like it as a type of amyloid disorders. It is the aggregates, not the high concentration of the amino acid itself, which kills the cells. This insight can put us on the path to a cure. If we can find a compound that disrupts the formation of those aggregates, we can prevent cell death and symptoms. Scientists in Gazit's lab, together with the Center for Personalized Medicine in Weizmann Institute of Science, are now screening thousands of compounds to see which has the potential to stop the amino acid from aggregating and offer hope for future treatment.
I hope it's now clearer. And just to end up, we always move from biology to technology, back to biology, and end up with just a story. So we saw the single amino acid story. I told you about our DNA-like structure. So we asked ourselves whether a dipeptide, which was our belief until then it's the smallest structure that can form all the assemblies, could form these such structures. And quite to our surprise, we could get the formation of very regular assemblies from a single protected PNA amino acid, and they can form also an ordered array. You see tens of thousands of spheres arranged on a very regular, physicist or chemist, Fourier transform. You see it's arranged in a hexagonal phase, each one facing 6 others. And one thing that we could notice just from the beginning is the very vivid color that you see when the structures are being formed. Sometimes you need luck. Just about the time that we wanted to publish it, there was a publication from another group in France, finding the mechanism which I mentioned in the beginning for the color change in chameleons. So the same nucleobase, G structure, guanine that I mentioned, that we found in a systematic reductionist non-biased way is used by nature, by the chameleons, to make the color. These structures are charged, so you have a repulsion between them. If you put more salt, it's masks the charge, so they are getting closer. And without getting anywhere into the physics or chemistry, we can do something like artificial chameleon, and actually we get now funding from this, from the US Air Force Research Laboratory and other. I'm in minus one.
You have 1 minute left.
1 minute left. So I hope that all of you heard Moshe Kohl's presentation in the first, excellent presentation in the first session. So he described this spontaneous formation of building blocks by the Urey-Miller. Sorry, I'm using another, but it's a similar idea. In a primordial world, you have the formation of these amino acids and nucleobases. And what we provide a model for the world. This was published already 10 years ago. When we discovered that just a dipeptides can form this structure, nowadays that we know that amino acids, nucleobases, can form ordered assembly, so you don't need the complex liquid that we mentioned yesterday or other. I think that we can understand how very simple molecules that could erase in a non-biological way, organized in a spontaneous self-assembly process to form the structure. Just to end up with how the science bon ton is changing over the years. This was published, as I said, December 2000. No, it's not now. So it's a mistake. It's 2006. So 10 years, and it got actually got neglected for almost 8 years. Now it's being rediscovered with the understanding that small structures are being formed. And I think that it provides us some interest, some insights about how life emerged. We did not invent anything. Da Vinci said all along that simplicity is the ultimate sophistication. I think that we gave a little bit of experimental evidence for this. You are now all experts of the periodical table, so you can see that our heart is in the field of chemistry. Although, as you could see, not all the elements in the same line are identical, just as you learned from Moshe Kohl. And if somebody is interested in this and other movies, you are welcome to visit our website and see it. Thank you.